Hanan joined us a year or two ago and came to us from a different background than what he's doing now in terms of the biology. So with us, of course, he's doing some cancer biology and glioblastoma. But in his past work, he studied how the coevolution of mammalian genomes with viral integration. For example, the long-term evolution. And we found that to be a very interesting mix, and we're pretty confident that could work well in a cancer biology setting, that the bioinformatics tools, the sequencing tools would all be very useful. And as well, the interaction between different components of a system would also come into play as the virus interacts with its host genome, just as the cancer cells interact with the normal cells in their environment, and these other aspects. And I think we found that to be a fruitful combination. So that's just an example of the way your career paths can go and how things learned in one area can be highly valuable in others. And I don't know if you'll see all the background of that in what he presents today, but I just wanted to introduce that aspect.
Okay, thank you so much. Hi, everyone. So today's actually the topic is not really related to the biology right now. So, but it's more like experience sharing with you. It's actually why I come out with this topic is actually because during the last 2 years we are facing a lot of issue or kind of mistake during for the project, and then as you can see, I refer all these things based on whole genome sequencing. So I'm thinking if I can share this experience to everybody, so maybe it will be helpful for everybody's project and make sure everybody can have a better result. So thanks for Tom's introduction. So let's go into today's topic directly. As you can, so basically for our project, we're using whole genome sequencing. So whole exome sequencing. The whole exome sequencing is a target enriched method to capture the specific region in a given genome. So for us, it's a human genome, and actually more specific, that should be a human cancer genome. So as you can see, it's really similar to the whole genome sequencing based on the next generation sequencing method. So the only difference is it's only sequence a specific region. For here is the exome, and then if you have a different method, but right now the most common method is the capture particle. So it's still based on the DNA. So DNA need to be fragmented. So DNA need to be fragmented. So sorry, the first thing we extract the DNA become fragment, and then we will have a primer, fob, or something they design or bait to capture the specific region we want to sequence, and then do the amplification. So the general method is similar to the whole genome sequencing. Okay, based on that, but the major difference is when we look at specific regions, you will see the whole exome sequencing enrich the sequencing reach to the exome region only. So that was actually how they designed the experiment. Here is a direct example showing from one of our sample. As you can see, this one is based on Illumina. The Illumina company actually designed the target for the UTR region, the exome, and then some each exome, but exclude all the introme. And then if we look at one of their example plot, you can see the ray coverage mainly cover the exome region, plus a little bit flanking region, and then a little bit of target region. So in the end, we will want to have something like this. When we have a specific genes, we know the coverage for their exome, and then we can do the somatic mutation calling in this exome region, and then we'll have the results. So based on based on these methods, I designed my own pipeline. So to detect the somatic mutation. So that will actually including the all the short variants, including short variants including the indels and then or single nucleotide variants. So as you can see, we have three major parts. The first part is the data preparations, and the second part is the mutation calling pipeline itself, and then we have a downstream mutation annotation pipeline. But in this topic, I'm going to talk about the major issue I'm facing here when I designed this pipeline. The pipeline has been is working pretty well today, but the issue we're facing is will be quite interesting. So actually, there are two major issues when we design the pipeline. The first one is the duplication. So this one is provided by the sequencing machine, or kind of say sequencing protocol. The second one is the mouse recontamination. And then we're also using a consensus site for mutation calling. Just say, I'm going to talk about why we're using consensus site later. But before I go into the details about the issue, so I'm going to talk about the project a little bit. So we are trying to research the glioblastoma glioblastoma from patient sample, and also based on the patient sample, we developed a cultured gliomosphere sample, and then also actually the patient cancer tumor directly, and then implant it into the mouse brain and to build a gliomopidox model. So actually, that's why we have the mouse risk contamination. So actually, you can see we got the patient sample, we have the cultured gliomospheres, and then we also have a mouse model here. So actually, we definitely the mouse cell contamination actually coming directly from the mouse brain. So that's actually because when we actuate the tumor, we are not only definitely we will try to make it as clean as possible. We'll actually have the tumor cell and plus a little bit mouse brains. So when we actuate them, so we do the cell sorting to remove all the non-tumor cells and then try to have the pure tumor cell in the end. But actually, the things is not going to happen that well. So it will never be that clean. So that's actually when we look at this data, you can see in here when the mouse contamination happen. Oh, so maybe this one will be better. So we actually do the mouse when we're finish all this process, we actually have the cell sorting method to make sure only tumor cell will come out and then go into the sequencing machine. But what really happened is when we try to, after the sequencing machine come out with data, so we try to analyze them, what we find is it's not 100% pure. Even we do the cell sorting, it's not going to reach 100% pure. So on this left panel, so you can see in the gliomosphere, so in the cell culture data, there's almost no mouse rays coming out. And then in the patient data, so suggests the same idea. So there is almost pure as possible because there are no mouse cell involved in any case, so it's pure. But actually, when you go to the xenograft, even we have the cell sorting results, but it's still not perfect. Some sample will actually reach roughly like 30% mouse rays contamination. So actually, you can see, and some of them were actually around 5 to 10% mouse ray contamination. So which means even if you finish the cell sorting, there's still roughly like 10 to 20% of mouse cells inside the tumor, or less. But when the sequencing machine do the PCR amplification, there's more mouse cell mouse rays will come out. So the left panel is a percentage risk, so it's probably not that clear. So then I transfer it to a log 10 scale. Then you can see there's some of them will actually become ambiguous. So the ambiguous risk is something not sure. So it's the risk, or is the risk can map to the mouse genome and the human genome. So I'm going to talk the why it happen like that. And then we'll actually can see human risk is always almost 100% or 99%. So in general, but the mouse risk, it will actually showing a little bit difference. And then actually you can see in here, so even in the glioma spheres and the patient sample, the mouse risk is not 0. So it's actually a little bit higher than, it's roughly like 0.1% or 0.01% in general. So why will this happen? It's actually because the nature of the mouse genome. So here is a quick paper to show why this is happening. So if we sequence the mouse genome directly and then do the whole exome sequencing using human exome capture kit to sequence the mouse genome and then capture it and map it to the human genome, you will see actually some mouse arrays can map to the human genome. Well, actually almost 73% of the mouse arrays can map to the human reference. There's only roughly like 30% cannot be matched. And then, so which means the mouse genome is really close, or at least for the exome level is really close to the human genome. And then if you look at this circle plot, you will see the race, or say the mouse array coverage is actually evenly or dispute. It's almost evenly dispute among the human genome. So every single region can have some mouse race, a really similar mouse race to match. And then if you go for the genes level, so definitely it doesn't have there's no exception for the genes. So it's not just like oncogenes or non-oncogenes. So all of them will have some mouse race. And then if we look at here, so you can see what really happened is, for example, we have one exome. There's tons of human race if there's a mouse array contamination. So there's tons of human race can map perfectly to its own locus, but some mouse array will actually become a contamination and then mixture together with the into the coverage. Okay, but how terrible it is when this thing happens. So here they actually, based on the paper, so based on the reference, they actually do a experiment to test the contamination rates with to test the relationship between the contamination rates with the somatic mutation calling number. It actually, you can see, even you only have 5% of a mouse race contamination, in general, you will have roughly like 20%. In general, they will have roughly like a 70% false positive results to come out from the mutation calling. So actually, you can based on this plot, that's actually based on the human race provide the property call, but the mouse array actually provide a fake or false positive SNP from their race, because these are the difference between human genome and then the mouse genome. So all of these mutation will come out as this for this 70%. So what truly happened in our data? So in our project, we're also using the mouse model. So before we filter the mouse array out from the sample, as you can see, when the mouse arrays, the number of the mouse array contamination going up, the raw mutation calling is actually showing a strong relationship between the mouse arrays and then the mutation calling. So you can see the mutation mouse array going up, the mutation we also have much more mutation in general. But when we do after we filter the mouse array out, you can the true thing is it's actually decreasing a little bit. But somehow we also still have some hypermutator. It may not be a good example, but if we look for the specific sample, you can see before the filter we will have something extremely high mutation calling, over almost 1,000. But when we finish the filter, there's only 30 or 40 mutation left. And then actually this one happened in general. You can see for each sample, almost every single every single after filter result decrease after the filter, decrease. So, but some of them actually will be quite pure, so there will be no change. So that's the mouse arrays contamination for the first part. And then we also have the duplication issue. The duplication issue is quite simple. So because the whole exome sequencing using is actually a massive PCR method. So as long as we do the PCR, there's a tons of PCR duplication will come out during the experiment. So especially when we also need to do the PCR amplification for the library. So we can expecting there's tons of PCR duplication artifact inside in the final data. So there's one actual example to show why the duplication is not good for in the end. So actually you can see if we have a normal or kind of a nature duplication events, so you will see results like this. You got we will have the heterozygous variance, we got 0 and 1. But when the duplication happen, the duplicates will actually generate 0 again. So actually we will have artifact 0 from we'll have an artifact 0 from the result. What happened here? And then well, actually that's definitely something we don't want to see. That's because you definitely don't want to know, okay, the mutation we found in the result is actually a fake result because the duplication happened. And then if we look at our data, so before, but also there's also an issue is if there's a lot of duplication, PCR duplication in the, PCR duplication in the data set, they will also have a high coverage issue in the end. So actually, the issue is the major issue we are facing today. So for example, we designed to have roughly like 2 million series in general for our experiment. But if our duplication level is really high, the available rates will actually decrease. So here is actually a back example for batch 9. You can see this is one of our batch. We designed to have around 20 million series in 20 million series in general. And then, but the duplication rate is almost 50. Then we have to remove 50% of the rates out. And then in the end, we only got roughly like a 10 million series available in the end. So this is actually what really happened. So definitely we don't want to pay more for the low experiment data, low quality data. But fortunately, most of the pipeline will actually have a deduplication method. So this is not a really high issue right now. Then the last one is we actually try to generate a consensus data set for the result. So that's why we're using. So actually, you can see in our pipeline we are using 3 different mutation caller for the mutation calling. So that's actually based on one paper. So it's published, it's part of a QCBio project. So in their method, they are actually using 4 different caller and then to generate a consensus. And then they actually have a paper to saying, if we're using 3 mutation caller together, we'll have the lowest false positive rate. Of course, you have more caller, then we'll have an even lowest false positive rate. But in here, it's actually more like a balance. The false positive rate is not the highest one, it's a reasonable, and then you actually reflect to the, you will have a higher true positive rate with a better result. So actually you can see in here, if we only using 1 caller, so we will have the false, or we only have the false positive and then true positive. But there, if there are 2 caller, there is some disagreement between them. So we'll have less false positive, but some mutation will disagree between two caller, and they will be removed. But if we go for 3 callers, so actually the false positive rate will go down quite a lot, and then although we have a more disagreement between each other, there's no large decreasing for the true positive. So if we look for the specific data, so you can look here for the TCGA, you can see actually for different mutation caller, the density of somatic mutation number, so actually quite similar to each other. So which means they don't generate really bad data. Sorry, they don't actually generate a really different data set between each others. So on the left, these 2 panels actually represent our own data. As you can see, the density is really close to each other's. If we go for the specific mean value for the mutation, the number of mutation calling for different pipeline, they're actually really close to each other. Although you will have a lot of outlier. So that's actually just because some sample will like hypermutators, so they have a much more mutation than the others due to the treatment, due to the drug treatments, or treatment, or kind of their own nature. And then also, in this case, we're using the consensus size. In the end, you can see even we generate the consensus size is the same as it's general. It didn't remove or kind of delete a lot of the sample, mutation from the pipeline. 3 pipeline actually, the general mean value of the number mutation calling is the same. Okay, so that's so actually that's the same issue we're facing before for the somatic mutation pipeline calling. And then the next one is some issue we are facing right now. This one is for the copy number. So in general, there is still using the same data preparation as the mutation calling, but you actually doing the copy number. Usually people doing the copy number by comparing the tumor sample with the normal sample. As you can here is an example, the example pipeline for how to estimate the copy number. So you usually begin from the raw data to calculate a bin level copy number, and then group all the bin level copy number into a larger segment, then showing the final result. The final result usually looks like this. So some people are probably quite familiar. So we will have some IG review. So you can see the red for amplification and then blue for the deletion. And also maybe some kind of a schema like this, still roughly showing the same idea. This is something finally we want to generate. But actually, there's some challenges for the copy number detections. So the first one is the GC content. This one is usually based because of the sequencing. Okay, this one is actually because of the sequencing method. So when especially for the platform, as you can see, when the GC content is really high, and then before correlations, high GC content region is actually even showing a good result. You will actually, the read depth is not even across the sequencing genome. And then after we correct this, So you can see the read depth for the sequencing exactly will be even. And then there's also got the multiple alignment issues. So some reads can map to the multiple region. Then you can see there are some peak for these regions. They are repetitive regions. And what really happens is if we have a deletion, for example, in here, the repetitive reads will actually go to this region and then fill the gaps. Then we will actually lose the deletion. And also there's an exome length issues. You can see when the exome into a certain the exome length in a certain region will actually have a much more risk than it will tend to have a much more risk than the other, than kind of a longer exome or shorter exome. And also we got a purity issue. Sorry, here's it may not be the best result, but here is actually showing you when we have a higher purity for the deletion, will actually show the deletion will actually more significant. But if we have some contamination or usually low purity result for patient, the deletion will not showing really good. And then also we have the cancer heterogeneity issue. So you can see when we do the single cell data, the copy number profile will actually quite different. It will show some different than the bulk tissue result. And also another issue I'm facing right now is actually we have the estimation for the region nearby the centromere and the telomeres. Here is a direct example from our project. You can see we got really high frequency deletion and amplification nearby the centromeres, which is here. So you can see in here. And then we actually found a paper there I mentioned when the copy number variation detection nearby the centromere, you will tend to have a really high frequency to find the amplification or deletion in general. And the last thing is actually the most annoying bit is there's no available tools can detect the copy number perfectly in any condition. So here is the 3 papers I found I reviewed before. So you can see in 2014 there's 4 common available or say for popular whole-exome copy number detection tool. They barely can draw any consensus with each other. So they are actually somehow all of them found some specific or kind of unique segment for themselves. And then after 3 years, things getting a little bit better, but you can see there's still a lot of disagreement between each others. And then even today, so in 2020, so you can see they still really hard to draw any conclusion between the pipeline, and that there's no guarantee which one is the better than the others. And that's because if you look for the comparison between the whole-exome sequencing tools with the array-based result, you can see they can see, for example, here, the bench is actually from array from the microarray for the copy number, and then the whole-exome sequencing data is actually didn't match with it. So you have the same one, but some tools will say this is a wild type, array says it's a deletion. Some of them will completely agree with each other, and then some of them will even against each other. And this one is from the reference. And actually I do the similar thing from the TCGA. It's a published data using a pipeline, using another pipeline. So when I selected the data is the copy number data is actually looks like this. There's 20 samples in here. We got some amplification and deletion in each way, in each genes. And then when we really look at the data in here, array-based and the exome-based, they don't match exactly. So right now it's almost 2021. So even after 4 years, so the tools didn't perform better this day. So this is something we are facing right now. So actually, you can see. Hopefully, this will the result I'm sharing will be helpful for everybody. So I think that's everything for today. So thanks for thanks for my supervisor and then Nick and Jennifer and Christopher for the data and the experiment. But yeah.